Hello, my dear friends. Welcome to my seminar. Let me start by saying few words about my own background. I'm Dr. Indrajeet Godke, and I've done my PhD from Department of Biochemistry, Indian Institute of Sciences, Bangalore. Since from 2015, I was working as a postdoctoral research scientist in an institute named IGBMC, which is situated in France. And currently, I am an assistant professor in Department of Microbiology, KTHM College in Nasik. So, as you can see on the screen, our topic for today is one of the most fundamental and critical process in cell biology, that is cell cycle. So this is the first seminar on cell cycle, and I'm going to begin with the events of the cell cycle and how they are controlled and coordinated. So for the next few minutes, I have divided my presentation in the following points. So the first, we will see the general overview of the cell cycle, and followed by that, we will see a detailed briefing of different phases of mitosis, and then we will discuss about the range of different techniques which are used in a research laboratory in order to identify cell cycle phases. And then we'll also elaborate on the Cyclin-CDK complex system, which is one of the important cell cycle control mechanism which ensures the cell cycle progression in one direction. And finally, I would like to conclude this seminar by explaining you the cell cycle transition which happens specifically during G1 phase of the cell cycle. So let's begin with the general overview of the cell cycle. So the classical definition of cell cycle is the series of cellular events that occur sequentially which causes cell to divide into two daughter cells. So this is a pictorial representation where the fate of a single chromosome is monitored throughout an eukaryotic cell cycle. So G1 is an intermediate phase between the birth of a cell following mitosis and the DNA synthesis phase. In G1, the cell actively reviews its environment and cell growth and ensure that the conditions are sufficient enough to support the DNA replication. So following G1 phase, the cell enters in the synthesis phase. So in synthesis phase, the chromosomes gets duplicated and then prepare itself for the next division. So in G2 phase, the cell actively grows with a lot of protein synthesis and prepare itself for the mitosis. So all the three phases, that is G1 phase, S phase and G2 phase together constitute an interphase. So end of an interphase, it marks the onset for the mitosis division. So during mitosis, lot of events happens which leads to the cell division. So at the end of the mitosis, the duplicated chromosome gets segregated into two daughter cells. So I would also like to draw your attention to one of the important phase of the cell cycle, that is a Z0 phase that we called as a resting phase. In G0, the cell is metabolically active but is unable to divide. So many of the neuronal cell, skeletal cells, and the liver cells, which are terminally differentiated Z0 cell cycle. And if you look at the cell cycle phases and their time duration, in a typical eukaryotic proliferating cell spends around 16 hours in the cell cycle. So of 16 hours, it spends around 15 hours in an interphase. So within interphase, the G1 phase, the cell spends around 5 hours. Whereas in S phase, where the synthesis or the DNA replication happens, the cell spends around 7 hours. Whereas in G2 phase, the cell spends around 3 hours. And the cell spends only 1 hour in mitosis. So within mitosis, which is divided into three different phases, that is prophase, metaphase, and anaphase, and telophase. So cell spends around 36 minutes in prophase, whereas in metaphase, it spends around 3 minutes, and in anaphase, it spends around 3 minutes, whereas in telophase, it spends around 18 minutes. So only during mitosis, we can see a duplicated DNA as in the form of a chromosome, which we can see on the screen. So then we can start about a brief overview of the mitosis. As you might already know that the mitosis is divided into different phases. So they are prophase, metaphase, anaphase, telophase, and cytokinesis. So the prophase is the first phase of mitosis which marks the onset of mitosis. So let's see what happens during the prophase cells. So the first thing which happens during prophase is the chromosome condensation. And the second thing which happens is the centrosomes, which are one of the important organelles to radiate microtubules, move towards opposite sides of the nucleus. And then it initiates the formation of mitotic spindle. And there is also a breakdown of nuclear envelope. So because of this nuclear envelope breakdown, this spindle microtubules attaches itself to the chromosomes. And they attach with the chromosome with the help of huge proteinaceous structure termed as kinetochore. And you can also visualize the cell under a fluorescence microscope. For this, you can label chromosomes and the microtubule with the differential color. As you can see here, the fluorescence micrograph showing the chromosomes which are red in color and the microtubule which are green in color. So followed by prophase is the second phase, which is prometaphase. And during prometaphase, the things which predominantly happens is the abrupt breakdown of the nuclear envelope. And now the chromosome can attach itself with the help of spindle microtubule at the centromeric sites. And you can also see under fluorescence micrograph these beautifully labeled green color microtubules and the red color chromosomes. Before we go into the detail of metaphase, I would like to draw your attention towards the process of chromosome condensation. So this is a model which shows some of the many levels of chromatin packaging which give rise to highly condensed form of mitotic chromosomes. So to begin with, the first DNA wrap itself around histone proteins. And the wrapping around histone protein gives a beads on a string form of a chromatin. And this results in a formation of 11 nanometer fiber. And this chromatin actually folds around itself, and that results in the formation of the nucleosomes, which results in a 30 nanometer fiber. And this nucleosomes again package itself in the form of chromatin fiber, which is in turn packaged into the chromatid of a chromosome, and which gives rise to a formation of 1400 nanometer fiber. So the net result of this process is that each DNA molecule has been packaged into a mitotic chromosome that is 10,000 fold shorter than its fully extended length. So because of all this condensation process, during metaphase, you can see the chromosome at the highest level of condensation. So let's look at what happens during the metaphase phase. So during metaphase, the first thing happens that the chromosomes are aligned at the equator of the spindle. And then the centrosome, which actually radiate the microtubules, and one of the microtubules, a kinetochore microtubule, get attached to the centromeric site with the help of kinetochore proteins. And you can appreciate this by looking at this beautiful fluorescence image where the chromosomes, which are red in color, which are equatorially aligned, and the microtubule, which are green in color, that are attaching to the chromosomes. So after metaphase, there is a phase called anaphase. So let's look at what happened during anaphase. So the first thing happened at the anaphase is the sister chromatid synchronously separate to two daughter chromosomes, and each is pulled slowly towards the spindle pole which it faces. And the kinetochore microtubule gets shorter and shorter, and then the sister chromatid actually moves apart. And both of these processes together contribute for the chromosome segregation. And you can see under a fluorescence micrograph that the beautifully labeled green color microtubules and the red color sister chromatid, which are segregated to two opposite poles. Following anaphase is the penultimate phase of the mitosis is telophase. So during telophase, the two sets of daughter chromosomes arrive at the poles of spindle and then they start decondensing. And also there is a formation of the new nuclear envelope around the reassembled sister chromatid. And then the division of the cytoplasm begins, and beginning of this division is marked by the formation of the contractile ring. And you can also see this under a fluorescence microscope. So the final stage of mitosis is cytokinesis. So the first and the foremost thing which happens during cytokinesis is the division of the cytoplasm. And this division occurs by the formation of the contractile ring with the help of actin and myosin filament. You can also see under a fluorescence microscope the formation of this contractile ring. And then the daughter cell receives one centrosome which duplicates prior to the next mitosis. And under the fluorescence microscope, you can see here the red color sister chromatids and the green color microtubule segregated into two daughter cells. So each chromosome contains a central constricted region called as centromere. So depending on the position of the centromere along the chromosome, the different chromosomes are designated. If the centromere is present in the middle, then it is called as a metacentric chromosome. And if the centromere is present between the middle and the end, it is called as a submetacentric chromosome. And if the centromere is present close towards the end, then it is called as an acrocentric chromosome. And if the centromere is present at the tip of the chromosome, then it is called as a telocentric chromosome. So depending on the centromere location, the chromosome has a different conformation during anaphase progression. So I would like to conclude the different stages of mitosis with the help of this video. So let's see this video all together. So during prophase, the chromosomes start condensing and the nuclear envelope breaks down. And the centrosomes, which are actually involved in formation of the microtubule, migrate to the opposite sides of the cell. And these centrosomes radiate the microtubule and then they attach to the chromosomes. During metaphase, the chromosomes are aligned at the equator and the kinetochore microtubule attaches to the chromosomes with the help of proteinaceous structure we called as a kinetochore. And during anaphase, the sister chromatid are synchronously pulled apart to the sides of the pole. And during telophase, there is a decondensation of the chromosomes and the nuclear envelopes start reassembling. And also there is a formation of the contractile ring and the two sister chromatids segregates into the daughter nuclei. So how we can identify different cell cycle phases in the laboratory? So here in this session, I am going to discuss with you the range of different techniques which we normally use in the research laboratories to identify the cell cycle progression. So the first and the very obvious thing to do is to look at them under a microscope. So here on the left hand side is the human fibroblast cells. So as you can see here, the cells are quite stretched. And this stretched morphology of human fibroblast depicts the cell which are normally in the G1 phase of the cell cycle. And you can also see some cells which are rounded. And these cells are nothing but these cells are specifically in the mitosis phase. You can also observe the yeast cell under a microscope and there is a bud formation in yeast cell. So the bud formation in yeast cell indicates that it has progressed towards the G1 phase of the cell cycle. So most of the cells which are unbudded are normally in the G1 phase of the cell cycle. And the second thing which we can use is BRDU labeling. So as you can see here, BRDU is a structural analog of a thymine. And the difference between the BRDU and thymine is just the presence of a bromine group. So what we do in the laboratory is we incubate BRDU with the cultured mammalian cells. And since the BRDU is an analog of thymine, it gets incorporated by DNA polymerase in the DNA. So once it gets incorporated in the DNA, we actually use an anti-BRDU antibody and by which we can identify the cells which have incorporated this BRDU. And the cells which have incorporated this BRDU are nothing but they are in S phase of the cell cycle. As you can see here by the fluorescence microscope, that the cells which are yellow or the green in color are normally the one which have incorporated BRDU, and they are actually present in the S phase of the cell cycle. Another way to look at the overall cell cycle population by using an automated sorting machine called as fluorescence activated cell sorting. And in this, we use a fluorescent dye. And this fluorescent dye binds directly to the DNA. And then the amount of DNA is actually proportional to the amount of fluorescence which is emitted. So here, the X and the Y axis depicts the number of the cells which are present on the y-axis, whereas the x-axis depicts the relative amount of DNA. So here, the 1 denotes the haploid content and the 2 denotes the diploid content. So cells actually fall into 3 categories. So the first is unreplicated component of the DNA, which is normally in the G1 phase where the DNA replication has not happened. And the second is the fully replicated component, which normally in the G2 phase or the mitosis, which happens followed by the synthesis phase. And there is a third part, which is an intermediate amount of DNA, which is normally during the S phase of the cell cycle, where the DNA replication has just begun or about to get completed. So from this graphical representation, what we can see is the amount of cells which are present in G1 phase are much more as compared to the other phases of the cell cycle. Whereas in the S phase, there is a least amount of cells which are present. And finally, we can also look at the cell by labeling the specific components of the cell cycle machineries. And the first component here we can label is the tubulin, which is a part of microtubule filaments and which we normally see during mitosis. And the second part we can label is histone. So histones are nothing but an integral component of DNA and which marks the chromosome, which are M-cherry colored. As you can see from this image that the chromosomes which are red in color are aligned at the equator. That means that the cells are in the metaphase phase of the cell cycle. And you can see the green colored microtubule. And this is a time-lapse imaging which we are collected by the fluorescence microscope. As you can see here, as the cell process from mitosis, specifically in the metaphase, the cell to migrate towards anaphase. And here, the sister chromatids are pulled apart with the help of the microtubule, which are labeled with tubulin, and the cells, the sister chromatid gets segregated, and the cell division occurs. So labeling with these components actually gives you the clues about the cell cycle phases. So with this technique, you can identify the cell cycle progression. However, there is a cell cycle control system which is present inside a cell which monitors at different transitory events during the cell cycle progression. So the first transitory event which happens is normally at the end of the G1 phase that we called as a start transition. So during this phase, the cell checks whether there is a favorable environment or not, and then it allows to enter in the S phase for the chromosome duplication. So second transitory point which happens is between the G2 to M transition. So during this, the cells check whether all the DNA is replicated and if all the environment is favorable or not. And if everything is favorable, normally it enters in the phase of mitosis. And the third transitory point is metaphase to anaphase transition. So during this phase, cell checks whether the chromosomes are aligned at the equator and they are attached to the spindle microtubules or not. And if everything is favorable, it allows to the metaphase to anaphase transition. And then cell subsequently enters into the cytokinesis phase. So one of the central component of the cell cycle control system is the Cyclin-CDK complexes. So we will look into the details of Cyclin-CDK system of regulation. So the activities of these kinases rise and fall as the cell progresses through the cell cycle, leading to cyclical changes in the phosphorylation of intracellular protein that initiate or regulate the major events of the cell cycle. Cyclical changes in the CDK activities are controlled by the cyclin proteins. So these cyclins are actually important to bind to the CDK proteins. So once the cyclin binds to the CDK protein, it results in the activation of the cyclin-dependent kinases. So there are 4 types of cyclins. Each is defined by the stage of the cell at which they bind CDK and functions together. All eukaryotic cell requires these 3 classes of cyclins. So the first is G1S cyclins, which activate CDK in late G1 and thereby help to trigger progression through start, resulting in the commitment to cell cycle entry. And their levels fall during the S phase of the cell cycle. The next is S phase cyclins. S phase cyclins bind CDK soon after progression to start and help to stimulate the chromosome duplication. So the S cyclin levels remains elevated until mitosis, and this cyclin also contribute to the control of some of the early mitotic events. And the third is M phase cyclins, which activate CDK that stimulates entry into mitosis at the G2 to M transition. M cyclin level falls in the mid of mitosis. And a separate regulatory protein complex, which is nothing but anaphase promoting complex, initiates the transition from metaphase to anaphase. So how do different cyclin and CDK contribute to the cell cycle progression? So cyclin and CDK complex associate with each other and then they phosphorylate range of different proteins. Those are important to initiate or to regulate the cell cycle progression. So in G1 phase, cyclin D and CDK4 associate with each other, and that results in an entry of the cell through the restrictive point. And followed by that, the stage is taken over by cyclin E and CDK2, which allow the cell cycle to pass through the G1 to S transition. Then during S phase, cyclin A and CDK2 associate, which regulates one of the critical events during the chromosome replication as well as in the G2 phase cycle. Whereas in mitosis, cyclin B and CDK1 associate together and they regulate the entry in the mitotic events. So this is a detailed structural basis of cyclin and CDK activation. So this data is generated from the X-ray crystallographic studies. As you can see here, when cyclin and CDK are important control points of the cell cycle regulation. So in an inactive conformation, normally there is no bound cyclin to the CDK protein. So because of this, the active loop, which is present at the roof of the active site, which we called as a T loop, actually sequesters the active site of this cyclin-dependent kinase. So once the cyclin comes and binds to the CDK protein, that results in the extrusion of this loop. And this results in the partial activity of the cyclin-CDK complex. And phosphorylation of this loop by the CDK activating kinase results the change in the shape of the T loop. And because of this, there is accessibility towards the substrate that result in the subsequent phosphorylation, which results in the regulation or initiation of cell cycle events. So the rise and the fall of the cyclin levels are important regulatory events with respect to the CDK activity. However, there are some additional events which regulates the cyclin-CDK complexes. So here, there is a regulation with respect to the phosphorylation and dephosphorylation mechanism. So once the V1 kinase, which phosphorylate The serine residues which are present at the roof of the active site results in complete inhibition of the cyclin and CDK complex. However, in presence of a phosphatase called as CDC25 phosphatase, result in the removal of this inhibitory phosphate moieties and thus activating cyclin and the CDK complexes. So in addition to phosphorylation and dephosphorylation, there are cyclin inhibitors which also regulate the cyclin-CDK activity. So once the cyclins are bound with the CDK becomes active. However, there are inhibitors of the CDK which in turn bind to the CDK enzyme and renders them inactive. So this is an example where P27, which is a key family member, binds to the CDK and cyclin molecules and it completely sequesters its active site. So once there is a tightly bound P27 to cyclin and the CDK protein, this results in completely abrogation of the cyclin CDK activity. So in a mammalian cell, there are different types of the KIP inhibitors which are present, and these are two different families. So first one is the INC4 family, which includes P15, P16, P18, and P19 protein molecules, which binds to the CDK4 and CDK6, thus rendering them inactive in the G1 transitions. So other family is a CYP or KIF family, which includes the protein P21, P27, P57 that binds to CDK2 cyclin E or CDK2 cyclin A proteins, and thus rendering the inactive transition at G1 and the SG2 phases. So let's get a bit deeper into the cell cycle transition, which is important during G1 phase of the cell cycle. So the proliferation of animal cells is largely governed by extracellular growth factors, and this growth factor actually binds to the cell cycle receptor and carry out the cell cycle transition. However, in absence of growth factor, the cells enter in a resting phase called as a G0 phase of the cell cycle. So once the growth factor binds to the cell surface receptor, it leads to the activation of the RAS GTPase. So RAS GTP is an important family of a signaling molecule which initiate a signaling cascade which leads to the activation through MAP kinase pathway. So once the MAP kinase pathway gets activated, it activates a series of transcriptional regulatory proteins. So the functions of these transcription regulatory proteins is that they pass it through the nuclear pore and enters in the nucleus and they bind to the DNA molecule and they regulate the expression of the immediate early genes. So one of the important immediate early genes is the transcriptional regulatory protein or the transcription factor MYC. So once this MYC get expressed, it also regulate the expression of the delayed response genes. So these delayed response genes in turn activate the G1 cyclin CDK, that is nothing but CDK4 and cyclin D. So once this CDK4 and cyclin D gets activated, it results in the phosphorylation of retinoblastoma protein. So this is one of the important protein which sequesters the E2F transcription factor. So as the transcription factor is actually sequestered by retinoblastoma protein, there is no transcription. So once the phosphorylation of retinoblastoma happens with the help of cyclin D and CDK4, that results in the release of E2F. So released E2F then binds to the promoter and regulate the transcription. So then retinoblastoma is also exerting its regulation at the level of CDK and cyclin complexes. So as the cell moves through the cell cycle and enter into early G1 phase, retinoblastoma phosphorylation declines and then again it reassociates with E2F transcription factor and this ensures the regulation with respect to retinoblastoma phosphorylation. And followed by that, so in the previous slide, we have just seen that the cyclin D and CDK4 activates the E2F transcription factor by phosphorylating its inhibitor, that is retinoblastoma protein. In addition to cyclin D and CDK4, there also exist another cyclins, which are cyclin E and CDK2. This cyclin E and CDK2 needs to be activated in order to cross the G1 to S transition. So this happens normally with the help of relieving inhibition. So another cyclin CDK complex which is important for G1 to S transition is cyclin E CDK2 proteins. But in a normal proliferating cell, these cyclin E and CDK2 proteins are bound by one of the inhibitors of the KIF family, which is P27. So there are two different mechanisms by which this inhibition is relieved. So the first mechanism includes the mitogen promoted growth signaling, in which the RAS-RAF make an ERK pathway stimulates the decreased synthesis of P27 protein. So as a result, there are no P27 molecules which bounds to cyclin E and CDK2 proteins. In the next step is carried out once the cyclin E and CDK2 becomes active, it carries out the phosphorylation of P27. So the phosphorylation of P27 results in its degradation. That result in activation of CDK2 and cyclin E molecules. So activated CDK2 and cyclin E in turn inhibits the anaphase promoting complex, which in turn prevents the degradation of cyclin E. So with this, I would like to conclude the first part of the cell cycle seminar. So to summarize the first part of this, we have seen in the first few slides the brief overview of the cell cycle. So which consists of the G1 phase, S phase, G2 phase, together we term as interphase, and then followed by that is a mitosis and the telophase. In the second few slides, we have seen different phases of mitosis, which include prophase, metaphase, anaphase, telophase, and the cytokinesis. Then we have also seen a range of different techniques which we have used in the laboratories to identify the cell cycle phases. And we have seen the detailed biochemistry of CDK and cyclin complex activation. And finally, I have concluded the first part of this seminar by explaining you the G1 to S transition. So in the next seminar, I would like to give you cell cycle transition which happens in M phase, and also will explain the cell cycle transition during synthesis phase. And also I will talk about the centrosome duplication and the checkpoints, which are nothing but the surveillance mechanism opted by cell in order to progress the cell cycle efficiently in one direction. So these checkpoints are included, which are P53 mediated checkpoint, ATM ATR mediated checkpoints, and anaphase promoting complex, that is the spindle checkpoint. And then I'll give you a brief overview of meiosis and the defect in the cell cycle progression and leading to the formation of tumors. Thank you.